Hi, welcome to the Bio-180 Tutoring Center videos. The topic of today's video is signal response or cellular response. So we have been talking about, in other videos, the reception or the receptor molecules that are involved in cell communication, and then the signal transduction pathways. And we've always just ended them with, oh, there's some cellular response. So now we're going to talk about what those actually are. Cellular responses tend to be some sort of regulatory thing. So there are three main types of cellular responses. The first is turning on or off genes, and this regulates transcription. As we know, proteins are extremely important for the function of cells and their environments. And so if we turn on or off genes, then it allows different proteins to be made to accomplish different functions. And again, by turning off these genes, it doesn't allow transcription or translation to occur, so the proteins are never even created. The second thing is that it can regulate enzyme activity. So if we've already created the protein, but we don't want it to function, or we want it to start doing what it's supposed to do, then we can regulate it. And we talked a little bit about different ways to regulate enzymes in another video, such as allosteric and non-competitive, and adding different molecules to activate and inactivate them. The last thing is it results in active movement of cytoskeleton or other elements. So sometimes the cell will get some response that needs to move in one direction or do something of that nature or divide, and so that's kind of under this last category. So that's basically it for the kind of responses that happen. But now we're going to talk about fine-tuning, or how we fine-tune, I guess that's why it's called fine-tuning, different parts of this whole process. So the first thing is amplification, and I've mentioned this in other videos, but in amplification we take one signal molecule, initial molecule, and we use it to create a ton of product or a large cellular response. And so an example of this is if you bind one epinephrine molecule to a liver cell, it will result in the creation of 10 to the 8th glucose molecules. And that's because what happens is one epinephrine molecule will bind, and it will allow for, it's a GPCR process. So by binding one epinephrine, it activates the GPCR, and while it's bonded, it can result in 100 G proteins being activated. And those 100 G proteins in turn will activate 100 adenine cyclases. And those 100 adenine cyclases, in that time that they're active, will be able to activate then 1,000 CMP, because each of them is activating about 10. And then those are able to activate even more, and it eventually gets to this really big number. And so that allows us to make a really big response. The next thing is specificity. So there's different types of cells. We have differentiation. A heart cell is different than a liver cell. It's different than a skin cell. It's different than a brain cell. So why is that? The reason is because even though all these cells contain the same DNA and the same genetic material, they differ in which genes they have active or turned on or off. And as a result, because they have different genes turned on and off, they have different proteins. And so when a molecule binds to a cell, it can cause a different response in one cell than the other, because they have different proteins, which result in different pathways being followed. And so an example of this is epinephrine. Again, epinephrine, when it binds to the liver, results in glycogen being broken down to glucose. Whereas epinephrine, when it binds to the heart, actually results in the cell contracting, which results in the heart beating faster. So that's an example of that. The next thing is signal efficiency. So a lot of things in the cell, we need to be having them happen fast, or else the cell won't survive. They need to be efficient in how they do things. And so this comes down to scaffolding proteins. You're going to need to know what this is. And basically, we have a lot of different enzymes and proteins involved in this pathway, and if we just have them all spread out, which they usually are, then it takes a while for one kinase to go find the other kinases that it's supposed to activate, and will probably become inactive by the time it is able to even find the enzymes it needs to activate. So instead we use the scaffolding protein. And so what happens is when a ligand or the molecule binds to the signal receptor protein, it will activate this scaffolding protein, which will already contain a bunch of the important kinases for the pathway. And so instead of having this kinase have to go out into the cytosol and find this kinase, they're already right next to each other, so they can already just activate each other and then lead to some cell process. So that's scaffolding proteins. The last thing is termination. This is essential for the cell. Make sure you understand this. I guarantee it's going to be on the test, because turning off a process, a cellular process, is just as important as turning it on. There's a lot of diseases that are a result of genetic disorders and that, or just chemicals that result in processes not being able to turn it off, and that can cause a lot of problems. So there's 4 different things we need to do to turn off or terminate a signal, and that is we need to release the ligand, we need to destroy the secondary messengers, we need to dephosphatase the enzymes involved in the process, and we need to do GTP hydrolysis. So let's go through all of these by looking at this example. So we're going to use cyclic AMP pathway, and what will happen is we have our signal molecule bind to our signal receptor, our GPCR, and that is going to in turn lead to the activation, because this is not activated, of the G protein. We are going to change its GDP to a GTP, and now that this is activated, it is going to go over to our adenine cyclase and activate it. And now that this is activated, it's able to create a bunch of cyclic AMP, and then this cyclic AMP is going to be used to activate kinase A, and kinase A is going to activate other things, which will result in a cellular response. So, how do we stop this cellular response? We don't want this to keep going forever. We want to stop this. So there's kind of a few things we have to do. First off is we can release the ligand. So we talked about KDs in other videos, and that's basically how tightly or loosely a ligand binds. And so this is only going to bind for a little bit. Eventually it's going to fall off. And so when it falls off, this GPCR is going to become inactive again. So that's great. This is inactive. It's not going to do its job anymore. But if this is still going, then it's going to cause all these things to still go. So we need to inactivate our G protein as well. And that already happens automatically, because every time after it has activated something, then it does hydrolysis on its GDP. This changes, or this falls off, and it takes up a new one, and it has now a GDP here, which means it is no longer active. Sorry, it loses the phosphate, it doesn't take it off. So our phosphate is gone, so now this is inactive. But if this guy is still active, then all this is still going to happen. So what happens next is we use one of our phosphatase, an enzyme that removes kinases. So if you remove the kinase from this, then it's going to be inactive, so now this enzyme is going to stop working. And so if we have other phosphatases being used to inactivate, then we're going to be able to inactivate kinase A with it. But if we still have all of our original cyclic AMP that we created, or a secondary messenger, then this is still going to be able to activate kinase A and lead to cellular response. So the last thing we have to do is we have to deactivate our CAMP, or cyclic AMP, or secondary messengers. And the enzyme that does this is phosphodiesterase. It takes our cyclic AMP, it changes to AMP, so it can no longer function. So we'll no longer activate kinase A, and we have stopped our cellular response. So make sure you understand 4 different ways that we terminate a signal. And also really important to know is the secondary messengers that, or the enzymes or proteins that destroy the secondary messengers. So with cyclic AMP, it's phosphodiesterase. With the IP3 pathway, it's a different enzyme. And with calcium, it's just the pump that pumps calcium back into the ER. So make sure you understand and know all of that. And I hope this video was helpful. That is the end to cell communication. It's very important for humans to communicate. It's very important for cells to be able to communicate and respond to their environment. Thank you for watching.